Welcome, everyone. So it's a pleasure to continue the Josep Carreras invited lectures. And today we'll be treated with a talk by Dr. Sonia Guedan. Sonia is a pharmacist by the University of Barcelona in Catalonia. And as a pharmacist, she likes drugs and new treatments, and in this way to address unmet medical needs. And what she has been doing in her PhD, postdoc, and now as a group leader. Her initial PhD was at the Catalan Institute of Oncology in Barcelona, where she developed an oncolytic virus that was able to degrade the cellular matrix of solid tumors. And this virus is under clinical investigations. This has been a very productive lab that produced many good tools and many good scientists. In 2010, she joined Dr. Carl June lab at Penn University, where she worked for 7 years. At that time, she helped to develop the first FDA-approved CAR therapy and to create a new CAR T-cell strategies and therapies that can be useful for other tumors and for other new clinical trials. In 2018, she was recruited by the Research Institute at the Hospital Clinic, that is called IDIBAPS, to lead the CAR T-cell program for solid tumors. And this, I'm sure, this has been a very challenging area. As you know, many difficulties, but I'm sure that this is the way to go. Many things start in leukemia and later move to solid tumors, from chemotherapy to cell therapy. And I'm sure this is going to be another avenue. So remember, there is a chat to ask your questions. You can write your questions in the chat. And the title of her talk is going to be CAR T-cell therapy, engineering immune cells to fight cancer. Sonia?
Thank you, Manel, for the introduction, and thank you, the Institute Josep Carreras, for the invitation to share my results today, and everybody for attending the seminar. So today we'll be talking about the CAR T-cell therapy. I will briefly explain what is CAR T-cell therapy, some of the clinical results with CAR T-cells for hematologic malignancies, and then we will focus on CAR T-cells for solid tumors, which is my area of expertise. So these are my disclosures. So CAR T-cells are a type of adoptive T-cell transfer, and adoptive T-cell transfer is a personalized therapy involving the administration of immune cells with anti-cancer activity to patients with cancer. So how can we get these immune cells that have anti-cancer activity? And today I will be focusing only on T-cells. We could also use NK or macrophages, but I will be focusing on T-cells today. So the first option we have is to get the T-cells from the tumors. This is what is known as TIL therapy from tumor-infiltrated lymphocytes. And the T-cell therapy is based on the observation that some cancer patients have T-cells in their tumors that are tumor-specific. And these T-cells can recognize the tumor, but for different reasons, they are being unable to eliminate the tumor in the patient. But if we can get a tumor biopsy and isolate these tumor-infiltrating lymphocytes from these biopsies, expand them in the laboratory, and reinfuse back to the patient, then these T-cells are able to completely eliminate the tumor and induce complete responses that are durable over time. So this therapy is working very well in patients with melanoma, and it's being now tested in other tumor types. It's personalized medicine. It's tedious, but the results are very good. But what happened for these patients or for these tumor types where we cannot isolate T-cells from the tumors? So we have another option, that is to get T-cells from the blood of the patients and then genetic engineer them to render them tumor specific by, for example, introducing transgenic TCRs or chimeric antigen receptors. So once we have genetically modified these T-cells, we can expand them in the laboratory and reinfuse them back to the patient. So where can we apply each type of therapy? Here we have a list of different tumors, and this is the number of mutations per megabase of DNA. So what we can see is here on the right, there are the tumors that have a higher tumor burden, while here are the tumor types that have a lower tumor burden. So the higher mutations, the higher number of mutations per megabase, and the higher more possibilities that one of these mutations will be immunogenic, and therefore these tumors with a higher number of mutations have more chances of having T cells that are tumor specific. By contrast, these tumors over here that have a very low mutational burden, for example, ALL, AML, or CLL, or neuroblastoma, glioblastoma, pancreatic cancer, these tumors that have a lower mutation burden have less options to have T cells that are tumor specific. Therefore, these tumors over here are the ones that have more chances to respond to TIL therapy or therapy with checkpoint inhibitors, while these tumors over here are the ones that have more chances, or that as they have less chances to respond to TIL therapy or checkpoint, then are good candidates to be treated with CAR T-cell therapy. So this is how typically adoptive T-cell transfer works. We start with apheresis from the patient, and then we isolate the required T-cell subset. Most of the clinical trials today just use bulk T-cells, a mix of CD4s and CD8s. Then we can stimulate these T-cells in the lab using bead-based antigen-presenting cells or cell-based, and then we redirect them toward tumor antigens using viral vectors or non-viral vectors. Typically, most of the clinical trials are using lentiviruses or retroviruses, and once we have these T cells that have been genetically engineered, we amplify them in the lab and we reinfuse them back to the patient. So what is a chimeric antigen receptor? So as its name indicates, it's a chimera, so it's a receptor that doesn't exist in the nature. We genetically engineer these CARs in the laboratory, and it's a chimera between a TCR complex and an antibody. So this is the CAR. It's made out of different modules. The extracellular part of the CAR is antibody-based, so it's a single chain FV that recognizes the antigens. Then we have a hinge. The hinge will give flexibility to the single chain FV, so it can bind to its epitope. Then we have a transmembrane domain. The transmembrane domain will link the T cell into the CAR into the T cell membrane. And then we have the intracellular domains that are the domains that are going to signal. So we typically have the CD3 zeta. CD3 zeta is the part from the TCR complex that signals once the alpha and the beta chain of the TCR recognizes the epitope presented by MHC molecules. The TCR complex signals through the 3D3 zeta. So this is what we include in the CAR, and then we include also the intracellular part of a costimulatory domain. That way, when the single chain FV recognizes the antigen, it signals through signal 1 and signal 2, and then the T cell gets fully activated to start releasing cytokines, proliferating and killing, and inducing the elimination of the tumor cells. So fine-tuning each of the models of that form a CAR is essential to improve T cell specificity, antigen recognition, and T cell function, and we will talk about this a little bit today. So CAR T cells have worked very well for hematologic malignancies. So there are 4 approved CAR T cell therapies for the treatment of CD19 positive malignancies, for B-cell leukemias and lymphomas. CAR T cells against BCMA for the treatment of multiple myeloma are giving very good results as well in clinical trials, and we will review them in a minute. So we hope they will be the next CAR T cells to be approved by the regulatory agencies. And finally, we have a panel of different CARs that are being tested in clinical trials, targeting C22, C20, CD33, CD123, ROR, or CD7 for the treatment of B-cell leukemias and lymphomas, AML, and TALL. So these are the 3 approved CAR T cells. So for Gilead has 2 different products, Yescarta and Tecartus, both of them are approved, but they use the same CAR, but they differ in the manufacturing process of the T cells. And Kymriah, it's commercialized by Novartis, and Lisosil was from Celgene was recently approved by the FDA. So all of these CARs have the same single chain FV. It's called FMC63, and it's from a mouse origin. And this is the part that will recognize CD19. They differ in the hinge, with Yescarta having C28, Kymriah CD8, and Lysocell IgG4 mutant. And then they all have the CD3 zeta, but they also differ in the intracellular domain and the transmembrane domain, being 41BB the most common intracellular domain for intracellular for hematological malignancies. Gilead have CD28, and then the transmembrane domains are usually either C28 or CD8. So Gilead uses gamma retroviruses, Novartis uses lentiviruses, and Lysocell, it's using lentiviruses. The main characteristic of this product is that they infuse a ratio of CD4, CD8 that it's 1:1, while the other T-cell products have variable percentages of these T-cells. So recently, a CAR that has been developed at Hospital Clínic of Barcelona, the RE01, has been approved by the Spanish regulatory agency for hospital exemption. So these CAR T-cells are now a product that can be administered to Spanish patients in Hospital Clínic. And this is very similar to the CAR from Novartis, but it differs from the single chain FV. And this is important because so far all the CARs that have been approved have the same single chain FV, so that recognizes CD19, while the construct from Hospital Clínic has a different single chain FV, and we have seen that the results that we are obtaining are very similar. And I think this is something very unique for hematologic malignancies, or for CD19 positive malignancies, because as we move to solid tumors and other malignancies, it's going to be very important that so the single chain FV is going to be very important, the affinity, as well as the different models for a CAR T-cell. So this is the clinical responses to CD19 CAR T-cell therapy for different diseases. For ALL, for pediatric ALL, it's around 80–90%. Our clinical trial at the Hospital Clínic of Barcelona, it's around 85% because it's mainly adult patients. Then for CLL, it's around 28%, and for cell lymphomas, it's around 50–60%. So in yellow, you have all the products that have been FDA approved and also approved by the European Commission. And here in green is the CAR from Hospital Clínic that has recently obtained the hospital exemption. So what we learned from these clinical trials that led to the proof of these CAR T-cell therapies? So first, we learned that patient preconditioning with fludarabine and cyclophosphamide, it's very important for the expansion and persistence of CAR T cells. So we need to precondition the patient in order for the CAR T cells to engraft in the patient and persist. We also understand now some of the determinants of response to CAR T cell therapy. So we know that T cells need to persist and expand, and we know that the composition of the infusion product is also important for responses. We need T cells that are young, that do not express inhibitory receptors, that are healthy and fit. We also learn about toxicity. So what we learn from toxicity is that it's usually on target toxicity. So it's because CAR T cells are doing their job. CAR T cells are recognizing CD19 getting activated and inducing cytokine release syndrome and B-cell aplasia. And this is because CD19 is expressed in malignant cells, but it's also expressed in healthy B cells. So as we treat patients with CD19 CAR T cells, we're eliminating both the healthy B cells and the tumor cells. And in some cases, we can also see some degree of neurotoxicity that we still don't fully understand why. Finally, we also learned some mechanisms of resistance and relapse to CAR T cell therapy. And what we know from, especially from ALL, is that when we have CAR T cells and we have the antigen, the CAR T cells are able to eliminate the antigen. So the resistance and the relapse mechanisms is because we either lose the CAR T cells or we lose the antigen in the tumor cells. So these are the results with anti-BCMA CAR T cell therapy for multiple myeloma. As you can see, the response rate, it's really high. Those are patients that don't have any therapeutic options. So we have mainly now three CAR T cells from Celgene, Janssen, and Novartis, and the complete response rate varies a little bit depending on the design of the clinical study, but it ranges from 18 to 68. So what we learned from these clinical trials with BCMA, that it's a little bit different from what we are observing in B-cell malignancies with that are CD19 positive, is that we have better responses when the patients are preconditioned. We have better responses at higher doses and also in less treated patients. But what we have also observed is that the most of the patients that achieve a complete relapse, a complete response, will eventually relapse to CAR T-cell therapy. So these excellent results that we are observing in hematologic malignancies justify the use of CAR T-cells in the more challenging setting of solid tumors. So the question now is, will CAR T-cells work in solid tumors? So compared to hematologic malignancies, solid tumors pose extra levels of complexity. CAR T-cells need to traffic to the tumor, infiltrate the tumor, and once in the tumor, they will have to recognize and kill tumor cells. So in order to fully eliminate solid tumors, CAR T-cells first will need to expand and persist in a tumor that it's going to be highly immunosuppressed. So in the case that T-cells are able to maintain their effector function in this immunosuppressive tumor microenvironment, they will have also to overcome the loss of heterogeneity of antigen expression that is characteristic of solid tumors. And finally, CAR T-cells are living drugs, and they are very potent, so we need mechanisms of control to avoid toxicities. These are some of the CAR T-cell trials in solid tumors. So we have now clinical trials for pretty much any kind of solid tumor malignancy. The most common CAR T-cells are those, for example, targeting GD2 in neuroblastoma and melanoma, mesothelin in lung, pancreas, mesothelioma and ovarian and breast. Also, there are several trials treating glioblastoma patients with IL-13 receptor alpha-2, EGFRB3, or HER2. And HER2 is also being targeted for sarcoma or breast cancer and head and neck. So these are the main antigens that are being targeted. Most of them are antigens that are overexpressed in solid tumors, but can still be expressed at physiological levels in healthy tissues. So we need mechanisms to make sure that our CAR T-cells are efficient but are not toxic, and this is the main challenge in solid tumors. So how can we, most of the clinical trials with CAR T-cells for solid tumors are showing pretty poor responses. We have seen complete responses in different clinical trials, but these are usually the minority of the patients treated. So in general, most of the patients, while we do see some type of anti-tumor effect, and we can see typically CAR T-cells getting activated, eliminating some tumors that are antigen positive, what we see at the end is that most of the patients do not achieve objective responses with CAR T-cell therapy. So we clearly need to increase the efficacy and the therapeutic index of CAR T-cells for solid tumors. And how can we do that? We need to take care basically of three different factors. We need to take care of T-cell intrinsic factors. We need T-cells that are fit, that are able to survive. We need to avoid tumor escape, and we need to control toxicity. And I will be briefly focus on each of these factors today. So in terms of T-cell intrinsic factors, one of the main mechanisms of relapse or resistance to CAR T-cell therapy is the loss of CAR T-cells or the lack of CAR T-cell engraftment. And this can happen because of a poor CAR design, because T-cells get differentiated and exhausted, or because the CAR is immunogenic. For example, if we are using a single chain RV derived from mouse T-cells, the immune system of the patient can recognize these single chain RV as strangers and eliminate the T-cells. So how can we enhance CAR T-cell fitness? So with a proper CAR design, choosing well our costimulatory domains, and I will talk about today, and using human single chain RV, optimizing the T-cell subsets by using less differentiated T-cells, and also using armor CARs, which means that we further modify the T-cells to express a CAR and a different transgene, for example, cytokines or costimulatory domains. So this is an example of what we typically see in when we use animal models. Our animal models are highly immunosuppressed. These are NSG animals with human tumors and are being treated with human CAR T cells. And we can see how when we treat, these are animals with ovarian tumors that express HER2, and we are treating them with HER2 CAR T cells. And when we use high doses that are not clinically relevant, we can see that all the tumors are eliminated. But as we lower the dose, we can see how some at the beginning, these tumors start responding to CAR T cell therapy, but at some point we lose the control of these tumors and these tumors relapse. So as these animals are immunosuppressed, so we don't really have a very immunosuppressed tumor microenvironment here. The main reasons why this can happen are because T cells are getting dysfunctional or because tumor cells are losing the expression of the antigen. And I will show you examples in both directions, but I will start for what we believe is T cell dysfunction. So when we analyze the tumors at the end of the experiment, you can see how these tumors express very high levels of HER2. So they are not losing the expression of the antigen. And these tumors are very highly infiltrated with T cells. Those are CD8 positive cells, so all these brown dots. So T cells are there, the antigens is there, but the T cells are unable to eliminate the tumor. And we believe that this is because while at the beginning they are functioning well, at some point they lose their effector function and they become dysfunctional. So how can we prevent this from happening? The first thing we need to take into account is that we need a proper CAR design. And CARs can be first, second, or third generation CARs. First generation CARs didn't have costimulatory domains that didn't work well in the clinical trials because they were unable to persist. With second generation CARs, the first generation CAR included the CD28 intracellular domain, and introduction of this costimulatory domain greatly enhanced the cytokine production of the CAR T cells and the anti-tumor effect. Then at Penn, they started working with the 4-1BB intracellular domain, which increases the survival of the CD8 T cells. So these CARs are not as fast as killing as the CD28, but persist longer. And then there are several other intracellular domains being tested. I have worked extensively with ICOS, and I'll show you some results today. And then we have the third generation CAR that include 2 intracellular domains. So what we know for sure is that second generation CARs work better than first generation CARs. We are not sure whether third generation CARs are better than second generation CARs, and we are not sure either which is the best costimulatory domain. And we don't know if there is a optimal CAR design or if each type of disease or antigen that we're target will require a different CAR design. So each of these costimulatory domains have its pros and its cons, and they need empirical testing in animal models. So during my years at the University of Pennsylvania, when I started exploring ICOS as a costimulatory domain, ICOS is a member of the CD28 family, and what is published in the literature for these molecules as costimulatory domains is that typically 4-1BB is studied in CD8 T cells and enhances the survival of CD8 T cells, while ICOS is typically studied in CD4 T cells and has a major role in the effector function of CD4 T cells. So what I hypothesize for this work is that maybe we cannot just use a CAR T cell for both CD4s and CD8s because those are 2 different types of cells. CD8s are cytotoxic, CD4s are the helper cells. So they might require different CAR designs. So in order to test this hypothesis, what I did is, and this is a bit complicated, I will try to simplify as much as I can, but we expand the CD4s and the CD8s separately, and we redirect the CD4s with different CARs containing a first generation CAR, the Zeta, or second generation CARs containing the CD28, the 4-1BB, or the ICOS, and did the same for the CD8s. And then at the end of the expansion, we treated animals with tumors with different combinations of CD4s and CD8s. So, for example, in these graphs over here, all animals were treated with the same CD4s. The CD4s and the CD8s were redirected with the same CAR, while here the CD4s were redirected with different CARs, while all the animals got the same CD8s. And here, all the animals got the same CD4s with the ICOS intracellular domain, and we changed the CAR that we used to redirect the CD8. So no matter which combination we did, if we analyze the persistence of the CD4 T cells, you can see here the red bars are CAR T cells that have the ICOS intracellular domain. So it doesn't matter which is the combination, CAR T cells with ICOS are always the winners in terms of CD4 T cell persistence. What happened when we analyze the persistence of the CD8 T cells? And I want you to focus here in this these animals. All these animals were treated with the same CD8 T cells. The CD8s have been redirected with a CAR that have the 4-1BB intracellular domain. But the persistence of the CD8s is highly dependent on the way we're redirecting the CD4s. So the best combination, if we analyze all the bars here, is CD4s being redirected with an ICOS intracellular domain, while CD8s being redirected with a 4-1BB intracellular domain. And we believe this is because the CD8, the 4-1BB enhances the survival of the CD8s, while ICOS makes CD4s very happy. And when we have happy CD4s, then we also have happy CD8s, because the 4s are helping the CD8s to do their job. So because this is a little bit, it's totally doable, but it's a little bit more complicated in terms of clinical translation, because it means you need 2 CAR designs, which means 2 different lentiviral vectors, expand CD4s and CD8 separately, and then mix them. We wanted to try what happens if we generate a 3rd-generation CAR that has both the ICOS and the 4-1BB intracellular domain in the same construct, so that both CD4s and CD8s are receiving these 2 signals, the ICOS and the 4-1BB. So what we observe here is these are animals with pancreatic tumors. You can see how the control T cells are growing. Animals treated with 4-1BB and ICOS 2nd-generation CARs get a stable disease. In red here we have animals that have been treated with CD4s with ICOS and CD4s with 4-1BB. And you can see how this is working really well at the beginning with this great anti-tumor effect. But then again, these T cells seems to lose the control of these tumors, and then we can observe this stable disease again. While when we treat these animals with the 3rd-generation CAR, we can see these complete responses that are durable over time. And when we analyze the persistence of these cells, we can see how both CD4s and CD8 T cells are persisting better when we have this 3rd-generation CAR combining the 2 intracellular domains. So this is a way where we can enhance CAR T cell therapy by optimizing the intracellular domains. And in the same direction, I'd like to show you, if you pay attention in this graph I showed you before, the combination of the CD28 T cells, they do not persist at all. You can see no matter which combination we do for both CD4s and CD8 T cells, when they are redirected with a CD28 intracellular domain, these T cells do not persist, although they do have a pretty good anti-tumor effect at the beginning. So because CD28 and ICOS are members of the same family, we thought that, and they signal through very similar pathways, so we hypothesized that maybe if we could modify the CD28 signaling so that it signals now more similarly to ICOS, we could enhance the persistence of this CD8. So CD28 and ICOS have a motif in common, that is this YMNM motif, that for CD28, for ICOS is YMFM. And this signal, this motif activates the PA3 kinase signal pathway through this Y motif, while the aspargine in the YMN motif activates the GRB2 pathway, and this is not happening in the ICOS. So ICOS can only signal through PA3 kinase, while CD28 signals through PA3 kinase and GRB2. So we hypothesized that maybe if we mutate the CD28 so that now it's more similar to ICOS, then we have a mutant that only signals through PA3 kinase and doesn't signal through GRB2. So this is one of the mutants we studied. We studied several mutants, but I want to focus on this mutant today. because this is a point mutation that makes now C28 signal more similarly to ICOS. And I'm not showing you, we did several, we analyzed these pathways, the GRB2 and the PI3 kinase pathways through Western blot, but I don't want to get into details. So you will have to believe me that now these C28 mutant signals through more similarly to ICOS than to C28. And when we analyze these CAR T cells in vivo animal models, we can see this is animals with CAPAN2 pancreatic tumors. These are the non-treated or the tumors treated with controlled T cells. They are showing progressive disease. These are the animals treated in white with C28, and you can see how these animals are initially responding to T cell treatment, but these T cells, this is day 21, so around here, these T cells stop persisting, and then these tumors end up coming back. Those are animals treated with ICOS. So ICOS CAR T cells are not as fast at killing as C28, but they persist better. So at the end of the experiment, the results that we get from C28 and ICOS are pretty similar. But if we pay attention to the C28 mutant, we can see how the C28 kills very similarly to CD28 at the beginning, but it's able to persist as much as ICOS. So by the end of the experiment, we can obtain complete responses in all animals, and these responses are durable. So to understand why is that this mutant was having a better anti-tumor effect than the C28, we analyzed we isolated T cells at day 15 from the tumors of these animals, and we observed that by day 15, C28 CAR T cells stop proliferating. This is by analysis of Ki67. We can see how these T cells are not proliferating anymore, while CAR T cells with the C28 mutant or ICOS are more actively proliferating in the tumors. So we also analyzed these T cells by RNA-seq, and you can find the details, all of this, in the paper we published at JCI last year. And you can see how the CD28 T cells look more differentiated and exhausted, while the CD28 mutant, that it's a point mutation but prevents T cells from signaling through GRB2, now this T cell seems less differentiated and less exhausted and more similar to ICOS signaling. So this is another example of how can we optimize these CAR T cell constructs so that we prevent exhaustion and differentiation and we enhance efficacy. Another option we have is to generate these armor CARs that I mentioned you before by genetically modifying the T cells to express other cytokines, and we can also use genome editing to delete genes that we believe that are detrimental for CAR T cell function. So if we can, for example, use genetic engineering with gamma retrovirus or lentivirus to express exogenous gene to enhance T cell functionality, avoid tumor escape, such as cytokines, as I mentioned before. We can use genome editing to delete endogenous cells that are detrimental for T cell function, such as PD-1. And then we have the option of combining these two strategies of knocking out the genes that are detrimental for T cell function while knocking in genes that can enhance the efficacy of CAR T cells. And this is something that I think is going to be very important in the field of the CAR T cells as we move to solid tumors or other diseases that are more challenging than to treat with CAR T cells and hematologic malignancies. So we will need these next generation CAR T cells that have been more genetically engineered. And this is an example of a paper that was published in Science last year for Dr. Carl June, which is a pioneer in the CAR T cell therapy. These are not CAR T cells. Actually, these are T cells that have been genetically engineered to express a transgenic TCR against NY-ESO. But what they did is they used the CRISPR-Cas9 system to eliminate the endogenous TCR and also to eliminate PD-1, the PD-1 gene. And by using this strategy, they could produce these T cells under GMP conditions and they treated 3 patients that they could not evaluate efficacy. These were patients with very advanced and refractory cancers, but they could show that this strategy was feasible, that the genome editing was feasible, and that the T cells that were genome edited could persist in the patient for months. So now this opened the window to start novel, new clinical trials testing knockouts, and we are studying this in the lab. I hope I can show you data soon. So the other thing we need to take into account is tumor escape. So we need to prevent loss of antigen expression, and we need to revert the tumor immunosuppression that we find in tumors. So in terms of tumor immunosuppression, you know that solid tumors are very heterogeneous in terms of the cell types. For example, we have tumor cells, but also we have cancer-associated fibroblasts, regulatory T cells, tumor-associated macrophages, and myeloid-derived suppressor cells, and aberrant vasculature. So we need to make sure that we eliminate or we transform these cells so that our CAR T cells will find a better tumor microenvironment to work. So we have basically 3 options to revert this tumor immunosuppression. One option we have is to generate CAR T cells that are specific against these cell types. So, and there have been CAR T cells published against that are able to eliminate cancer-associated fibroblasts or tumor-associated macrophages. The other option we have is to modulate the tumor microenvironment, and we can do this by using these armor CARs that secrete cytokines such as IL-12 or IL-18 that are pro-inflammatory cytokines, and that will polarize these immunosuppressive T cells towards pro-inflammatory T cells. In this, also as modulation of the tumor microenvironment, we can use combination of CAR T cells with other drugs that could change the tumor microenvironment, such as oncolytic viruses, and I will talk about this in a minute. And finally, we can confer CAR T cells with resistance to immunosuppression by, for example, what I just told you, using a genetic disruption of inhibitory receptors such as PD-1, so that the T cells are resistant to this immunosuppressive tumor microenvironment. And this is a huge area of research in CAR T cells for solid tumors. So the other thing we need to prevent is the loss of antigen expression. And this is something we have seen already with hematologic malignancies such as ALL, with CAR T cells targeting CD19. In the case of Kymriah, they typically see around 20% of the patients that are losing that are having tumor relapse that is CD19 negative. So when they analyze which kind of mutations that these B cells have, they found different mutations in CD19, and the different mutation in all these patients end up with a protein that is unable to be attached to the cell membrane. So all these B cells now, they all have different mutations. All these patients have different mutations, but all of them end up with a loss of CD19 expression. And CD19 is a marker of B cells. So basically all B cells, all malignant B cells are gonna have this CD19 marker. But in solid tumors, this is way more heterogeneous for the antigens we are targeting. So if we are losing CD19 in B cells, in solid tumors, the chances that we lose the antigens are much higher. So this is an example of solid tumors that we see in the lab. Those are solid tumors that express mesothelin and are treated with CAR T cells against mesothelin. When we analyze the tumor at the end of the experiment, we can see how the percentage of mesothelin expression is much lower in CAR T cell treated tumors than the non-treated tumors. And you can see clearly here in this immunohistochemistry how are we losing the expression of this antigen. So how we can prevent that. So one strategy we have is to use dual CARs or tandem CARs. Dual CARs is when we introduce 2 different CARs in a T cell, while tandem CARs are one single CAR construct that have 2 different single chain FB. This is being tested in hematologic malignancies However, so for solid tumor, this is a little bit, it's being studied and it's very interesting, but as we increase efficacy by targeting 2 antigens, we can also increase toxicity. So we need to be very careful with this therapeutic index. So another option I proposed during my years at the University of Pennsylvania was to combine CAR T cells with oncolytic viruses. So the idea here is that when we treat tumors with CAR T cells, they found a very immunosuppressive tumor microenvironment. However, if we first treat this tumor with oncolytic viruses are a danger signal that our body is very used to fight against. So by introducing an oncolytic virus into a tumor, first the oncolytic virus, it's an anti-cancer therapeutic, so it's going to do some tumor debulking, it's going to start eliminating the tumors, but at some point the immune system is going to realize the virus is there and it's going to infiltrate the tumor to eliminate it. So by doing this, we can revert a little bit this immunosuppression and creating a more pro-inflammatory tumor microenvironment. And then the oncolytic viruses have the advantage that can also be further modified to express cytokines, chemokines, immune checkpoint inhibitors, or BiTEs. So that it can also partner with CAR T cells to fully eliminate the tumor. So in a project we did in collaboration with Ramon Alemany from the Institut Català d'Oncologia, we combined CAR T cells with an oncolytic virus that expressed a BiTE. BiTE are similar to CARs, but it's 2 single chain together that one binds to the tumor antigen, the other binds to CD3. So these molecules, these antibody-based molecules, put in touch T cells with tumor cells, and they activate T cells so that they eliminate the tumor. So in this project that I want to show to you, we have CAR T cells targeting folate receptor in combination with an oncolytic virus that has anti-tumor efficacy by its own and that expresses a BiTE that targets EGFR. So we have 3 different mechanisms of action, 3 different therapeutics here. And you can see these are animals with colon cancer tumors. You can see how non-treated tumors grow. In this case, the oncolytic virus didn't have anti-tumor effect by itself, although this is kind of strange. Usually for most of the tumor types, oncolytic viruses have some type of anti-tumor efficacy. Those are CAR T cells. Again, you can see how at the beginning the CAR T cells are able to control tumor growth, but then the tumors end up escaping. While we combine this therapy, we can prolong the survival of these animals when compared to the CAR T cell treatment alone. And this is something that we are hoping to start a clinical trial at the University of Pennsylvania testing this combination therapy. Finally, and very briefly, we need to control toxicity. Now we are preventing toxicity. Now we are treating toxicity. Ideally, we should be preventing toxicity. So currently, we are treating toxicity with administration of tocilizumab, with non-pharmacological immunosuppression, or by modifying the CAR T cells to express suicide genes so we can selectively terminate these T cells in case of toxicity. This has been shown to be very efficient in animal models, but while it's being tested in clinical trials with human patients, it has not. We still don't have reports showing that these suicide systems work in patients. The other option we have is to prevent toxicity. The best way to prevent toxicity is to target antigens that are expressed only in tumor cells and not in normal tissue. Unfortunately, this is not the case for most of the antigens we are targeting. The other option we have when targeting shared antigens that are overexpressed in tumors but expressed also at lower levels in healthy tumor, is to use affinity optimized CAR T cells. And this is what I want to show you today. And then we have also the option to use non-integrative CAR expression as RNA electroporation. In that case, the CAR will be expressed only 3 or 4 days, and then the T cells will not express the CAR anymore. So we would need to give repetitive doses of these CAR T cells. And finally, there are the next generation of CAR T cells that are more sophisticated CAR constructs that can mitigate toxicity. Today, I want to show you some results with an affinity optimized CAR. This is a low affinity CAR that targets HER2. It's trastuzumab based, but has been mutated. So now we have a low affinity antibody that can discriminate between HER2 expressing normal at normal levels or at high levels. And in order to test this CAR, we generated this model of heterogeneity of expression, antigen expression, in we have a triple negative breast cancer cell line that doesn't express HER2, and then we genetically modified this tumor cell line to express low, medium, or high levels of HER2. And we combined, we treated these different cell lines with CAR T cells, with our low affinity CAR T cells against HER2. This is a cytotoxicity assay, and you can see here that the cells that do not express HER2 or express low levels do not die when treated with CAR T cells. By contrast, when we treat these CAR T cells that express medium and high levels of HER2, then with CAR T cells we can see this clear killing even at low doses of CAR T cells. We tested this in vivo as well, just using these two cell lines, the one that expresses low levels of HER2, that would be physiologic levels of HER2, and these lines expressing high levels of HER2. And we can see that only the animals with tumors that express high levels of HER2 are responding to CAR T cell treatment, while animals respond with low levels of HER2 do not respond to CAR T cell treatment. So this would indicate that these CAR T cells could be a safe option to treat patients, but it also highlights the fact that we might also have tumor escape due to overgrowth of tumor cells that express low or negative levels of the antigen. So we always need to balance this efficacy and toxicity, and we are now developing ways to optimize this CAR so it's safe but can also eliminate tumors that express low levels of antigen. And with that, I would like to thank the people in my group at IDIBAPS, the clinicians at the Hospital Clinic that were very close with us, Álvaro Urbano, Manuel Joan, Aleix Prat, Julio Delgado, Valentín Ortiz, the people from the University of Pennsylvania, as most of the experiments I presented today were done at the University of Pennsylvania, and also the collaborators at IDIBAPS that developed the oncolytic virus and the fundings as well. So thank you very much, and I will take any questions you might have.
Thank you very much, Sonia. Great talk, really a lot of data and very interesting results. So I'm trying to put together the questions here. So let's start this one here. Yeah, so I have myself first one general question about cards and commercial cards right now, okay? And because you stressed out the cooperativity between CD4 and CD8, okay? So what are given to the patients right now in ALL, they really take care of the fraction of CD4, CD8, how it works, how relevant is that? Is there a difference there in the commercial products?
Yeah, there are differences. So yeah, Gilead started with PBMCs, with Yescarta, and Novartis started with T cells, but it's bulk T cells. So at the end of the expansion, some patients have 90% of CD4s and some others have 90% of CD8. So it's very heterogeneous. And that's why Celgene decided to change this protocol so that they expand the CD4s and the CD8 separately, and then they infuse them together to the patient at 1-to-1 ratio. And with that, they have not proved this is better. What they claim is that now you have a product that is more similar for everybody, and it's easier now to understand, to find correlation between efficacy or toxicity between patients, because otherwise it's very difficult to understand why some patients are responding and some others are not when you have such different products. So for our CAR at the Hospital Clínic of Barcelona, the Prodigy starts with 50% of CD4s and 50% of CD8s, but this is how the manufacturing starts. But by the end of the expansion, again, we have a very heterogeneous product with some patients having 90% of CD4s and others 90% of CD8. So this is something that you will need to optimize.
And do you think to compensate this to finally have a product that infused that is 50%, 50%?
So in animal models, it has been shown that CD4s together don't work well and CD8 together don't go well. And when we mix them at 1-to-1, then this is always the best option in animal models. But I have not seen preclinical studies when they mix 60 and 40 or 30 and 70. So what we know is that we need both players. We know we need both of them, but I don't know what's the best ratio.
And related to the tumor escape by the loss of the antigen there, so I understand that there are many mechanisms, and there are some tumors that predominate one and the other, depend on the CAR. It doesn't depend on the CAR.
It does depend on the CAR actually, because not all CARs show the same percentage of escape by loss of antigen expression. For example, with Kymriah, there is a 20% of patients that lose CD19 expression, while in our CAR T-cell products at hospital clinic, this percentage is much lower. So it might be depending on how long the CAR T-cells persist, but it also might depend on the disease and the plasticity of these tumor cells to mutate and to lose the expression of the antigen. And this will for sure get worse as we treat solid tumors.
Yeah. So I understand that one of the successes of in the leukemia, in acute lymphoblastic leukemia, was itself the CD19, the antigen. So are there any other CD19 molecule, or we have to compromise for molecules that are expressed only in a few tissues? Because I know you mentioned mesothelioma, for example, only in the so there are other CD19s out there, or I know people have been looking, but there are others there.
Yeah, so Manuel, first I would like to go back to the previous question, because one mechanism by which tumor cells could lose antigen is by methylation, by epigenetic changes. So any idea you might have to to prevent this loss of antigen expression, it's very welcome. So in terms of, CD19, it's an ideal antigen because it's homogeneously expressed in cancer cells. And it's only expressed in B cells, and we can survive without B cells. So, but there are not that many antigens as CD19. BCMA is a pretty good antigen, and in terms of solid tumors, we have the EGFR-B3, which is a mutant from EGFR that is pretty tumor specific, but is typically not expressed in 100% of the tumor cells. It's usually expressed in around 20-30% of the tumor cells in glioblastoma patients. So we have nothing else as CD19 as of now. And yeah, finding good tumor antigens is the main challenge in the field.
A question for the other here. The difference between CAR T cells and CAR natural killer therapies. What is the main difference, Gordon? What is the best and worst?
Yeah, well, they are using different cell types, right? And NK cells, we don't have that much experience on NK cells, and we'll have to optimize the CAR construct, because the first therapies with NK cells would use the same CAR construct as that is designed to activate T cells. So now NK cells are being designed with CARs that have the DAP12 molecule that are more adapted to NK cells. And the main difference is that NK cells do not persist well, and they cannot be frozen, as far as I know. But you can use them you can have universal NK cell products. So you can get the NK cells from blood from normal donors and then use them to other patients. So this is a great advantage that we can have over NK cells over T cells, that they can be universal.
Yeah. So what you mentioned is, again, this is very hot area of the holy grail of the shelf without personalized CAR T cells. So what is that? There are many companies. I know there are many trials. So what is the status of that?
Well, of course. So CAR T cells now are a highly personalized therapy. So if we could move to universal CAR T cell, it would be ideal. So there are several trials testing CAR T cells where we knock out the TCR and the beta-2 microglobulin. So we knock out the TCR so that T cells don't induce graft versus host disease, and then we knock out the beta-2 globulin so that the HLA is not expressed and the patient cannot reject the CAR T cells. So the thing is, there are several companies trying this. We are not sure how well this is going to work, and at that point, we still do not know if CARs are cooperating with TCR to signal. So it might be that CAR T cells need the TCR there to properly function, because we believe that the TCR is somehow included in the CAR synapse. And so we really still don't know what happened when you eliminate the HLA or the TCR from a T cell. Is the CAR going to work the same way? So these are things that are under investigation right now.
We have here another NK CAR aficionado here. Do you believe that they will overcome CAR T cells?
I believe both CAR T cells and NK CAR cells will have a role in the future of cancer therapy. So I am confident that we will have some NK CAR products approved. I don't know if they will overcome T cells. I have never worked with NK cells, so.
Okay. And in a similar line, what about true CAR cells using natural TCR approach?
So this means CARs that recognize molecules in MHC class 1.
Yes.
Yeah. So, well, that would be ideal. That would be ideal because then you are targeting tumor-specific epitopes. It's challenging, but it would be a way to use a CAR that overcomes all the problems of MHC that some tumors have of lack of co-stimulation, for example. But it's obviously challenging. There are, I think, more than 10 papers exploring this approach. So we are getting there, but that would be great. Yeah.
Thank you. Another question: it will be possible to combine CAR T-cell therapy with autologous dendritic cells, pools with lysated tumors, to increase specificity of the CARs?
Well, this is a very good question, actually. It's the first time I've ever get this question, but I have to say that targeting a single antigen in CAR T cell is challenging. We will have tumor escape. So we need to activate the immune system, the endogenous immune system. We need to activate the endogenous T cells of the patient so that they help the CAR T cells to fully eliminate the tumor. So this means we need to induce an epitope spreading so that CAR T cells cooperate with endogenous T cells and the endogenous immune system to eliminate the tumor. And I think that using the dendritic cells, it's a great way to find a cooperation and to induce this epitope spreading that we definitely need in solid tumors.
Thank you. Another question here. The increased PI3 kinase pathway activation in ICOS CETA CAR T cells also increased T cell cytokine production, or just increases proliferation?
So the PI3 kinase signals through an increase of phosphoic AT that increases the survival of the T cells. In terms of proliferation of cytokine production, we have an increase on PI3 kinase, but we have a decrease in GRB2. So at the end, we don't have an increase in cytokines. The cytokine level are pretty similar, but always the C28, typically the C28 is the intracellular domain that releases higher amount of cytokines.
I think it's later going back again to the 2 questions ago. Could TRACs, the 4th generation CARs, be a way to improve efficacy in solid tumors?
Yeah, TRACs are the same as, well, it's what we know as armor TRACs, but armor TRACs are CAR T cells that have been genetically modified to express any transgene. TRACs are more focused on expressing cytokines. And I think for solid tumors especially, it's going to be very important for the same reason as I just say, a way of inducing epitope spreading, to have these TRACs that express IL-12 or IL-18, and these and are able to turn this tumor immunosuppressive microenvironment in a more pro-inflammatory tumor microenvironment, and to induce this epitope spreading that we need.
Almost the last one, because I don't want to take a lot of your time, and we promise to stay within an hour. Neoantigens, like a target. Neoantigens in a wide way. So what do you think? Instead of seeing...
So targeting neoantigens is great, and it can be done with TILs or with TCRs. It's a very challenging strategy, but the problem with neoantigens is that they are typically different in each patient. So it would be great if we could identify a neoantigen that it's expressed in different patients, because otherwise you need to identify the neoantigen, identify a TCR, clone the TCR, and treat the patient. And I hope the technology is gonna improve fast, and that maybe in a near future this is feasible and is fast. But this would be the safer way to treat a patient with genetically engineered T cells, to treat a neoantigen. And then the same as I said before, we can also treat neoantigens with CAR T cells that recognize the neoantigen bound to the MHC class 1, develop a single an antibody, but it's a long process and very highly personalized.
Yeah, I think that there are many targets that has been discussed, and many cell types that you can use here. Just to finish with a question that to the real world of the hospitals. So what do you think is going to happen with the price of this product? Because one of the problems is really if this can be sustained by the health public system. This is going down. It's going to be down very soon. Or now we have limited cases that you can use it. So still a system can allow to use these drugs, this approach. But what's going to happen?
No, that's problematic as if CARs start working well and you start using them even as earlier lines of treatment and in more cancer types. It's true that the prices are very high, the actual prices. It's a one-time treatment, so it's only one dose. And if it really works, it will prevent also the health system for expending a lot of money for patients that end up relapsing. But I hope it will end up, the prices will end up going lowering. As also we need to improve the manufacturing system, and if we could have universal CAR T cells, this would definitely lower the cost of the T cells. Also, if we could use, for example, DNA instead of lentiviral vectors, that this also increases a lot the price of the manufacturing process. So hopefully we'll find a way.
And just a curiosity, do you see any difference between the lentiviral and the retrovirus? Is this here?
Not in T cells. So in previous studies in CD34 positive cells, they did see that with retroviruses, sometimes the CD34 cells would end up inducing leukemias, but for T cells, we have never seen a T cell becoming leukemic because of a retrovirus. So there are not that many studies comparing the effects of lentivirus versus retrovirus in T cells, but we know that both of them so far has been safe, and there have been patients being treated with CAR T cells with retroviruses since the 80s, and we have never seen a transformation. So I think as of now, we don't see major differences between these two viral vectors.
Thank you very much, Sonia. It has been really great. We have learned a lot, and a lot of food for thought, for sure. And please join me to congratulate the speaker. Thank you very much.
Thank you.
Thank you, Sonia.
Okay, bye-bye.